pleasure to present our next speaker, Dr. Todd Michael Cooper. He's an attending physician at Seattle Children's Hospital and a professor of pediatrics at the University of Washington School of Medicine, and Evans Family Inwood Chair in Pediatric Cancer. Dr. Cooper is also the director of the Pediatric Leukemia Lymphoma Program and co-director of the High Risk Leukemia Program. His interest and expertise focus on the development of new therapeutic strategies for treatment of children with acute leukemia. He serves as a chair of the COG Acute Myeloid Leukemia New Agents Committee, and he's leading COG new study AML 1831, which is a phase 3 study for newly diagnosed children with acute myeloid leukemia. He's also a clinical trial lead for Leukemia Lymphoma Society funded master trial for relapsed pediatric acute leukemia. So Dr. Cooper will talk to us about updating the risk classification in pediatric child with myeloid leukemia over the next half an hour. Dr. Cooper.
Thank you so much. I hope you can hear me well. I can't begin to tell you how honored I am to be asked to present to this Congress. I really, really appreciate it. I'm happy to be able to talk about pediatric AML. The speakers thus far have been outstanding, focusing on childhood ALL, and I look forward to talking to you about updating the risk classification in pediatric AML and the Children's Oncology Group experience as it relates to the current phase 3 trial, and also have a couple of slides at the end that introduce this Leukemia Lymphoma Society pediatric master trial for relapsed acute leukemia as well. So first, I will give very brief background, very brief, because I think most people on this wonderful Congress already know this information. Unfortunately, we are lagging behind our colleagues in the ALL world in terms of overall survival. And one of the reasons here is our therapies really are largely unchanged, unfortunately, even since the 1970s, when it was first introduced, the 7+3 of cytarabine and anthracyclines. We have made some strides, but these strides have basically been because of maximal intensity of chemotherapy, because of stem cell transplantation, and because of we're getting better with supportive care. I do have listed in this slide the inclusion of a targeted therapy for AML that's now FDA approved in the United States, gemtuzumab, the CD33 antibody, and that has resulted, as I'll show you in 1 second, in some improvements. So this is a Kaplan-Meier survival curve of the North American studies since really the 70s. And you can see here that there have definitely been, because of clinical trials and everything else we discussed a second ago, there have definitely been improvements. One of the biggest of which was right here, the 2891 study led by Bill Woods, which did intensive timing chemotherapy. And you can see here that was quite a leap. However, we instituted a MRC backbone here, an AML 03P1, which was the pilot study for gemtuzumab, and 0531, which was the phase 3, which also resulted in improvements. But our last clinical trial, AML 1031, really did no better, and in fact might have been a bit worse. I don't have time to talk about why, but in general, there's a few reasons, and one of which is we de-escalated the cytarabine quite a bit in AML 1031 by skipping our last intensive cytarabine cycle. So obviously we need to do better. And I'm going to focus just on one way that we can do better, and that is assigning patients to the correct therapies. Before I do so, I do want to briefly, very briefly, go through these last two COG phase 3 trials and point out two things. Number one, you can see increased complexity between the AML 0531, which incorporated gemtuzumab, and the AML 1031 trial, which incorporated bortezomib into the standard backbone. But also, this trial did test a FLT3 inhibitor, sorafenib, which is accepted for publication, and these results have been outstanding. What I also want to point out on this slide is the relative simplicity of the risk classification, which you can see here and here. And basically, the high-risk features had not changed over these two prior phase 3 trials. The low-risk features hadn't, and really the only difference was, rather than having 3 risk stratifications in 0531, there were 2 in 1031, and that is due to the incorporation of minimal residual disease by central flow cytometry into this. So, in contrast, we see the current phase 3 trial, of which I'm the chair, and I say that because it's almost apologetic in a way, because it's an incredibly complex study, which a lot of that surrounds the risk assignment. But this particular clinical trial randomizes patients between a backbone of cytarabine and daunorubicin plus Mylotarg versus liposomal cytarabine and daunorubicin and CPX-351, also known as Vyxeos, plus Mylotarg. In addition, this also allows for incorporation of a FLT3 inhibitor, not only for our FLT3 ITD patients with a high allelic ratio, but also, as I'll describe in one slide a little bit later, we've incorporated the addition of gilteritinib, which is that FLT3 inhibitor for both of these arms, for FLT3 activating mutations that are not driven by the internal tandem duplications. And importantly, what we'll focus on mostly is this risk assignment that occurs after the first cycle of induction chemotherapy. So in order to understand our risk classification, we have to understand that, and I think everybody on this Congress knows, that childhood and pediatric AML are not the same. They are biologically distinct, and we have continued to make this case both nationally and internationally to our regulatory authorities and also to our pharma colleagues. And what we've been able to determine is that the younger you are in pediatric AML, the more likely you are to have an AML that is driven by fusions rather than mutations. And this is the top panel here is from Catherine Tarlock and Sohail Mashinchi. This has since been published, but this is based on Foundation Medicine results from clinical trials and is retrospective. And then we also see here in this panel the adult TCGA whole exome and genome sequencing versus the target AML, which was whole genome sequencing as well. And what you can see here again is a much higher prevalence of the fusions in children versus the mutations in adults, such as your IDH mutations and the DNM2As, etc. In addition, from this target initiative, we also found new fusions and other abnormalities that are driving pediatric AML that have terrible outcomes, which we have listed here: are CBF GLIS2 patients, our ETS fusions, our KAT6A fusions, and also our NUP98 fusions as well. And not just with our KDM5As and NSD1s, but as will be published very soon. I think it's been accepted for publication, US and European collaborative, showing that really any NUP98 fusion does quite poorly. In addition, I wanted to show you this, this contrast from adults, whereas the tyrosine kinase domain mutations that we often see in adults, the TKD mutations, the D835s, are present in adults, and they are present in children as well. However, in contrast to adults, we also see other tyrosine kinase and non-ITD AML driving mutations as well. And you can see here on the right panel, Drs. Mashinchi and Tarlock also added crenolinib to patient samples, and we are seeing that these mutations do respond to FLT3 inhibition, and this is why we have incorporated this into our pediatric trial. So I wanted to give you the rationale or some of the lead up to this change in risk stratification. So what we did is we went back to these last 3 phase 3 studies within the Children's Oncology Group, and we did central review of clinical karyotyping in FISH, our molecular alterations, and then used the target data as well as individual RNA-seq and qPCRs for specific fusions to find additional risk stratifying lesions. And what we did, and this isn't universally accepted, but we basically said any of these fusions by which had an event-free survival of less than 50% is high risk. And what we did was, and you remember how simple that risk stratification was in the last 2 phase 3 trials. Now look at it. There's a number of new fusions and mutations that are added to this list, including our MECOMs, our DEC-NUPs, which we often see with our FLT3 mutations, our KAT6As, FUSE-ERG, GLIS2, and this is a biggie, and this is what we're seeing a lot of on the current phase 3 trial. There are a number of KMT2A mutations that have been added to high risk, and these patients do extremely poorly. In addition, the MLL-T10s with any partner gene do extremely poorly as well. Obviously, our FLT3 ITDs with high allelic ratio, our RAM phenotypes, which go with our GLIS2, and then really our MRD positive patients as well. Our favorable prognostic markers really have not changed much. But before introducing this into the next phase 3, we went back to the last 2 phase 3s to see what sort of impact this would have. This was presented at ASH, and manuscript has been submitted. And what you can see here, if we apply these criteria to the retrospectively to the 0531 study, before adding MRD, 31% of the patients were high risk. After entering MRD, 40% of the patients were high risk. And you can see in contrast to the 13% that were high risk on the actual study. And then obviously the survival curves are sussing that out and showing that these patients do extremely poorly. We wanted to validate that, and we did that by going back to the AML 1031 study. And you can see with the addition of FLT3 and MRD to 1031, 30% of those patients were high risk on the actual protocol. And when we put these criteria into the 1031 retrospectively, 41% were high risk. So putting it into practice, we were able to move these high risk criteria through protocol development, and these high-risk criteria are now incorporated into the study. There are 3 risk groups. Those patients who have favorable prognostic cytogenetic and molecular markers with negative minimal residual disease get 4 cycles of chemotherapy, because even on that last trial that didn't do as well, those patients that had these features actually did quite well. Our high-risk patients, which now is a much larger group, receives stem cell transplant. It says here 3 cycles of chemotherapy, but we're allowing patients to go to transplant after 2 cycles of chemotherapy. And then everybody else receives 5 cycles of chemotherapy, basically per the arm of AML 0531. So 5 cycles of chemotherapy. So we have 3 risk groups once again. Now, this is where I think that this is one of the great things about this trial, is that this diagnostic testing, while quite complicated, is actually wonderful because there are eyes on every piece of this centrally. So obviously sites are doing their local cytogenetics in FISH, but this is reviewed centrally by our team of study hematopathologists and cytogeneticists. In addition, a sample is sent to hematologic, so central flow cytometry review at diagnosis. So they are performing baseline flow cytometry so that they can accurately have that baseline moving forward for their end of induction 1 and 2 responses. And then they're also looking at the RAM phenotype, which thus far has gone along perfectly with our GLIS2 fusions detected by Foundation Medicine. In addition, patients are sending, the sites are sending samples to the Biopathology Center nationwide, so centrally, where they are extracting DNA and RNA, and then they're sending it to a number of other laboratories for further interrogation of these samples. One is sent to Fred Hutchinson Molecular Oncology, which is centrally reporting the FLT3 ITD with allelic ratio testing. They're also the source of truth for the study for our NPM1s and BZIP-CEPB alphas. And then another sample, both of the RNA and the DNA, is sent to Foundation Medicine, who first, within 10 days, are reporting those FLT3 non-ITD activating mutations back to the sites. And then second, they're reporting their RNA and DNA sequencing, both in a study-specific report and then also a CLIA laboratory report back to the sites. And you can see here, here's one of a couple of different schemas of how we're incorporating everything. You can see molecular oncology, Foundation Medicine, local cytogenetics and FISH, and hematologics are all reviewed. And one of our other study committee members, who's very computational, Matthew Cutney, helped to put together this AML risk matrix. And this is all automated. So either the laboratories for 4 out of 5 of these are directly entering this data into a variety of case report forms, or the site, in the case of the Foundation Medicine activating mutation, are reporting this into a case report form. And then what happens is you have a centrally automated risk group assignment, which then goes back to the site and tells them they're either LR1, LR2, or high risk. And that is done by the end of induction 1, with the addition also of your minimal residual disease. And so again, this is all reviewed centrally by all of us and then forwarded to the site. There's sometimes a lot of back and forth, but it has worked out incredibly, incredibly well. And it's something that we're all very proud of thus far in this study. And you know, if you go back and look at what we projected on the past 2 phase 3 trials to be the number of high-risk patients, thus far, we're very, very close to that. So 43% of these patients are high risk, 37% are low risk 2, and 19% are low risk 1. So, so that's mostly our risk stratification for newly diagnosed patients, but even so, and we hope that we're getting those patients to the right therapies and we're intensifying therapy via transplant for more patients, and hopefully we'll get better outcomes. But still, we expect that a large number of these patients are going to relapse. And as we all know, these patients need better approaches, and we all need to come up with a way to all work together for these patients, and also have longitudinal data collection from diagnosis to relapse. And we hope that this can be moved forward by this initiative that we're all very excited about, the Leukemia Lymphoma Society Pediatric Acute Leukemia, or PEDAL initiative. They did this in adults, called Beat AML. This to me is a much more complex thing, because this is going to be a collaboration between the United States, North America, Europe. Also, we expect Australia and New Zealand to participate in this. And what this is it'll be the first comprehensive international study that will very systematically collect information and provide data on targets for children with relapsed leukemia. And I'll show you how we're going to do that in just a second. But this is quite important. It's very, very difficult to accrue rare subsets of patients onto a trial if we're doing it piecemeal. We have to come together to be able to do this nationally and internationally. And through such of an effort, and with the support of Leukemia Lymphoma Society, it will be great to bring our pharma partners to the table so that they'll have a mechanism to accomplish this. And hopefully we'll bring these drugs to children earlier. And also our regulatory authorities are involved in this, and prospectively we're running everything by them, because we want these companies to be assured that they'll get the information they need for approvals and fulfill their regulatory obligations. So the backbone for this in North America is this what we call a screening trial. And the way this happens is if you have a patient that so you put this screening trial through your IRB. There's no treatment on the screening trial, so it should go through fairly quickly. And if you have any newly diagnosed patient, or any relapse patient or any suspected relapse patient, and that's very important because these are the patients we're missing. What's happening in real life is that we suspect a relapse and we do a marrow, and only then do we start to try and figure out what trial they should go on. And what we want to collect is these suspected patients earlier. So confirmed or suspected patients can enroll on the screening trial. They submit specimen. They also submit via case report form a lot of other clinical data. And then these samples will either go centrally to hematologics, which will confirm the relapse and report the blast percentage. And they also will report sub-trial target markers. So there's sub-trials associated with this screening trial for which these patients can be assigned. The blood or bone marrow will also go to Foundation Medicine, and those patients with greater than 10% blasts will be sequenced, and these will also go into the database. And what you can see here, this is another very large schema on how this will work. We'll have all this data from 1831, so we'll be able to connect this longitudinally to relapse. Patients go on the screening trial. Most of these patients will have been on the phase 3, but we're collecting those that aren't, and their clinical data. All their data from Foundation 1 and hematologics will go into this tool that's been developed by Sam Bolchenbaum and his group at University of Chicago, which will take all this information and tell patients what trials they're eligible for. We are not assigning them, at least not in North America, because that hasn't worked in the past. I don't have enough time to go into it, but there's already 5 or 6 trials that are pretty close to being activated. And the screening trial has already been approved. So we're very, very excited about this. And what Leukemia Lymphoma Society has done is linked the Children's Oncology Group Network and also what they're calling the UPAL Consortium, which is a number of existing consortium throughout Europe that we're really excited about. And so we're working together not only on these screening trials, but also on international trials moving forward. So we're very excited. So in conclusion, AML still has an overall poor prognosis, devastating side effects, and we need to move forward, like our ALL colleagues, with more targeted therapies. The current phase 3 utilizes a risk stratification based on standard techniques as well as next-gen sequencing, and that it's a very coordinated process using central review that will hopefully assign patients to their proper therapies and those that need more intensification, such as transplant. And then finally, the LLS PEDAL initiative. I just wanted to introduce that to everybody here because we're very, very excited about it for our relapse. And it's not just AML, it's ALL as well, certain subsets of ALL. And we're excited about that trial as well. So again, there's a very, very large number of folks that we need to acknowledge here, both from PEDAL, Leukemia Lymphoma Society, COG, our European colleagues as well, and also our regulatory authority colleagues, and also our pharma partners as well. So thank you so much for the opportunity to talk to you. I really, really appreciate it, and I'm honored. Thanks.